introduce me, my name is Eugenio Raymond, and I'm going to present the research I carry out at MIT in Ireland using molecular tools for the quantification of bivalves and decapod larvae from field samples. First, the reason for choosing these organisms were their economic relevance, being estimated their contribution to the Irish national economy in 397 million euros only in 2020, and their ecological function, taking part in important biochemical cycles, deeply affecting the trophic webs, modifying their ecosystem, and being considered in some cases as keystone species. As many other benthic organisms, bivalves and decapods present a larvae phase, which is responsible for the distribution and viability of the adult population through their spat abundance and location. However, little is still known about it. This gap of knowledge is explained due to three main interconnected issues in the monitoring programs studying zooplankton. First, the large amount of samples required for a proper coverage for these studies. Then the common group classification as larvae without any further specification, and the identification method relying on morphology, being the latter the limiting factor, since the decapod larvae present several development stages that can be really similar between different species, as well as for bivalve larvae that are almost impossible to differentiate. This makes the identification really time-consuming and dependent on the research expertise. Hence, the current monitoring programs quantifying these organisms are not able to do it in a proper time frame, of course. To cope with this limitation, several emerging approaches have been proposed. However, the real-time PCR or qPCR is the one with better perspectives. This technology relies on the classic PCR reaction of the naturalization, annealing, and extension, combined with specific assays which only amplify the target DNA and fluorescent dyes. These dyes, combined with the double strand of DNA, produce high fluorescence. So, after each PCR reaction, more DNA is generated and more fluorescence can be recorded. The high throughput qPCR uses this change in fluorescence together with a standard curve of a known amount of DNA to determine the initial amount of DNA present in the samples. In the same way, the digital PCR uses the same approach, but without need of a standard curve, since with this method only one molecule of DNA is expected to appear per each PCR reaction. With this background, my research aims to test the capacity of the high-throughput qPCR for the quantification of bivalves and decapods from field samples. To do so, a cross-validation with the morphological approach will be carried out, as well as the descriptions of the sample composition for these taxas. For this study, the samples were collected in the east coast of Ireland in 5 different sampling stations, taking 5 replicates per station through vertical tones with a plankton net. Each sample was split into 2 tubes, one tube preserved with Lugol, another tube preserved with ethanol. The tube with Lugol was used for the morphological identification, and the tube with ethanol was used for the genetic analysis. The morphological identification was carried out with using taxonomic keys and microscopes, completing the analysis of all the samples in 3 weeks. The organism counts were transformed to organism densities, and due to the normality of the data, a non-parametric test was used to test the difference between and within stations. For the genetic analysis, the DNA was extracted using the PowerSoy Pro kit by QIAGEN, and the qubit fluorimeter was used to estimate the total amount of DNA present in the sample. As Madalena already introduced some things about the biomark, I will complete the description. In both cases, for the digital PCR and the high-throughput qPCR, the reactions were carried out in the biomark. If you watch the video, that device that appears, the transparent one, is the chip where the PCR reaction occurs. And the big machine is the biomark, where the thermal cycle is set and where the change in fluorescence is detected. In the digital PCR, the chip used was a 37K IFC, and after dilution testing, 3 assays were selected, targeting marine invertebrates, bivalves, and decapods. And a Pearson correlation was used to compare the digital PCR and the microscope outputs. On the other hand, the high-throughput qPCR were carried out using the 9696 IFC, and 24 species and 2 genus were targeted using 45 assays. The standard curve were set with 5 tenfold dilutions of an oligopool, and the efficiencies of the assays were calculated. The output of the non-parametric test showed a significant difference between stations and a not significant difference for their replicates. As you can see in the NMDS plot, there was a cluster for the station 1, 4, and 5, and the densities were plotted per station, obtaining the total zooplankton, bivalve larvae, decapod larvae densities. Same approach for the digital PCR output, obtaining the estimated amount of DNA copies per target for each station, obtaining the marine vertebrates, bivalve, and decapod genetic signals. Same for the qubit output, plotting the nanograms of DNA per microliter per station. From the 45 assays used in the experiment, only 17 show good performance. So their efficiencies were calculated and the amount of DNA estimated. In this figure, the output of the experiment is presented, plotting the log transformation of the amount of DNA copies per species per sample, being grouped in a station and showing as well the oligopool signal. In this figure, the yellow represents more copies and the blue represents low amount of copies. And finally, bivalves and decapods were grouped together per station, being the bivalves classified in mussel, clams, scallop, oyster, and razor clams. The cross-validation showed, using the correlation, showed a positive correlation for bivalves and decapods, but not for marine invertebrates. This difference might rely on the wide number of targets of the assay, since the large difference between taxa may interfere in the quantification. The only relevant difference for bivalves appears in the station 1, and as previous authors already proved how the change in size or age of the larvae in bivalves doesn't really affect the amount of DNA, this difference may rely only in a human error during the pipetting. And in the case of the decapods, the absence of genetic signal in the station 3 and 4 might rely only in the absence of this organism in the sample, since as you can check in the density plot, the maximum of decapod were less than 2 organisms per cubic meter. So it's quite likely that they were not present in the sample. About the sample composition, the assay performance was not ideal. In the literature, an ideal efficiency is described between 90 and 110%. However, almost all of our assays had an efficiency above that. In the literature, this is explained due to the amount of DNA present in the sample that can create some kind of inhibition. This inhibition can affect the quantification of a small amount of DNA, as it was the case for the assays for Spizula solida and Nicora puber, that were not able to quantify the small amount of DNA from the oligopool. Also, the large efficiencies can produce quantification in late PCR cycles. In the literature also, this is described as false positive results. And it was the case for Magallana gigas and the NCIS species assays. Although it would be expected that the digital PCR and the high throughput PCR will have a similar pattern, they present a different one. Of course, the exclusion of some species during the data mining, as well as different target gens, might have an effect in the quantification. However, it seems that the inhibition is the main cause of this difference. when we check the total amount of DNA, we can see how the station 2 and 3 are the ones with less DNA, and actually are the ones more similar between each method. And finally, about the sample composition itself, you can see how almost the 90% of the bivalves were mussels, being the rest clams and scallops. And in the case of the decapods, were swimming crab and green crab, being the latter less than 1% in the total amount of DNA. At this point of the presentation, I wanted to make like a break to highlight how amazing these results are. I used to spend a lot of time behind a microscope during my studies, and I know how time-consuming and annoying the quantification of the zooplankton can be. And these results were obtained in one afternoon. 25 samples were screened in one afternoon, obtaining results at the species level, and not a species like qPCR, species bivalves that are almost impossible to differentiate with a microscope. If we try to do the same with the classic morphological approach, it will take weeks and only will be possible if you are a proper taxonomist specialized in differentiating these organisms. So to conclude, I'm going back to my objectives. Of course, there is room for improvement. However, this research already showed the outstanding perspective of the high throughput qPCR for the quantification of these organisms, as well as their capacity to cope with the limitation of the current monitoring program quantifying them. Also, there was a positive correlation for both methods, and we were able to describe the species composition, the sample composition at the species level. As I say, there is room for improvement, so the next steps could be test the assays again after correct the dilutions, and keep working in the assay panel in order to cover all the relevant species. These are the references used in the presentation. My acknowledgement, especially to Luca and Dennis, and thank you for your attention.